students?
So we do have research students. We have students from high school as well. I'm in high school myself, and we have professors and our panelists from a wide age groups. Right.
Yeah, I apologize in advance. It's going to be targeted more to the kind of undergrad, postgrad research level. It's a bit hard to target all those levels at once.
No need to apologize, sir. I mean, I'm interested in learning more about, the undergraduate level study.
Great. And how many people are you expecting? Now I can't hear you. I've got my volume up, but I still can't hear you.
So I was saying that we're expecting around 100 people.
Great.
Yeah.
And is it mainly been from India, or have you had attendees from outside of India?
So I wouldn't say particularly from India. We have attendees from other countries as well, students in other countries. So yeah.
Great.
Sir, I hope everything is fine in Australia in terms of COVID right now, by the way.
Things are really good, actually. We're lucky because we're an island country, but we've also had a very good public health response. So Melbourne you might have heard that Melbourne had a second spike, but it only went up to about 800 cases a day, but very good response to that. Now for the last 30 days, we've had no cases at all in Victoria. And so we're very lucky in Australia. I know it's been tough in India. I've got some students from India and I know how hard it's been there.
Yes, sir. I mean,
is it getting better in India? Is it?
Honestly, where I'm seeing it's not getting better because people aren't taking precautions anymore. They're just taking it. But yeah, in certain parts of India, the situation is definitely improving.
Right.
But also I'm like just so super glad to hear that like at least some parts of the world people are taking the proper precautions and people are at a safe place relatively.
Yeah, look, I think it demonstrates if you follow the public health advice, you can effectively eliminate it. That's what Australia's demonstrated and New Zealand as well. It's much easier if you're an island country, obviously, where you can control your borders. That makes it easier. But anyway, the vaccines are coming, so hopefully, but it's hard, how do you vaccinate so many people so quickly? It's very challenging.
Yes, sir. But at least we are a bit hopeful that things will get probably better sometime soon.
I think so. I think with vaccines coming, but I think people shouldn't rely on vaccines. I think the public health measures are still really important because it will take a very long time to vaccinate, the proportion of the population that would eventually provide herd immunity.
Yeah, totally agree. And I mean, I think if I'm not wrong, even when we do start vaccinating, we would prioritize the ones that need it first. So essentially, a lot of people would have to wait in the beginning.
Yes. And healthcare workers will be the first in Australia, for example.
Yes, sir. Also, sir, I'm extremely sorry, but we'll be starting, I hope on time, not before that, because since we're expecting for a lot of people to join, I hope that's fine.
Yes. So is that 7 minutes from now?
Yes, sir, just 7 or 5 minutes more.
Yeah. No, whenever you just you start when you want to start. I'm ready. And also tell me when to share the slides, and I'll do that as well.
Definitely, sir.
And thank you, Anushri and Stuti. I just saw a message from Anushri. So thank you very much for the invitation.
Thank you so much for joining, sir. This is Stuti here. I cannot turn on my video right now, but I know that you have been extremely accommodative despite your busy schedules, and there had to be some changes made, but it was really amazing to accommodate you. And yeah, and we're so happy that you're here, and we really look forward to your talk. It's a bit early in India. It's 11:53. So as Jasmine said, we will be starting in another 5 to 6 minutes. So we're just hoping that more people would join. And also with regards to the participation, we definitely do have high school. We have an assortment of people. So we have high schoolers, we have early career researchers, we have postdoctoral students, we have masters and undergrads. So it's completely an assortment. So I understand that it might be a bit difficult to cater to a particular audience, but at the same time, we've had an amazing response from all our speakers as well as from our attendees in terms of the talks that have happened. So I'm sure that we're really excited for your talk, and it will be an amazing one. Thank you for joining us today.
Well, thank you. Thank you again for the invitation.
And personally, sir, I'm truly looking forward to the nature versus nurture talk and its possible intersection with neuroscience.
Great. Excellent. Thank you.
Professor, I think we can start now, if that's fine with you.
Yes.
All right. So, hey everyone, let me welcome you all to the last day of the Neuro November 2020 convention. And I think I speak for most of us when I say that this event has been truly insightful and cleared a lot of misconceptions about neuroscience and a lot of other fields, particularly in the sciences. So my name is Jasmine Singh, and I'm currently in the 12th grade and extremely passionate about learning what goes into our daily decision making and psychology and everything else related to neuroscience. I'm someone who's just wanting to explore. I'm also the head of content at Stimulus and been working with them for a while. And I think, like in this event, honestly, from understanding our own laughter to the way that one dances and to how rats possibly help each other, I think this event has given us a lot to think upon and learn from, honestly. So we have our first speaker for the day, Professor Anthony Hannan. He is a National Health Medical Research Council's Principal Research Fellow and head of the Epigenetics and the Neural Plasticity Lab at the Florey Institute of Neuroscience and Mental Health at University of Melbourne. His talk is titled Nature, Nurture and Neuroscience, Gene-Environment Interactions Mediating Experience-Dependent Plasticity in the Healthy and Diseased Brain. And as I mentioned, I'm truly looking forward to the nature versus nurture talk and its possible intersection with neuroscience. So Professor Hannan received his undergraduate training and PhD in neuroscience from the University of Sydney. He was then awarded a Nuffield Medical Fellowship at the University of Oxford, where he subsequently held other research positions before returning to Australia on an NHMRC RD Wright Career Development Fellowship to establish a lab at the Florey Institute. So he and his colleagues provided the first demonstration in a genetic animal model that environmental simulation can be therapeutic, which has eventually led to new insights into gene-environment interactions in various brain disorders, including the Huntington's disease, dementia, depression, schizophrenia, and autism spectrum disorders. And what I think is truly interesting is that his research team explores how genes and the environment combine via experience-dependent plasticity models of specific neurological and psychiatric disorders, which involve cognitive and affective dysfunction, investigated at behavioral, cellular, and molecular levels so as to identify pathogenic mechanisms and novel therapeutic targets. So Professor, I would love for you to just have the virtual stage that we have here today and begin.
Yes, thank you. It's wonderful to be here. Thank you very much. And I'll just share my slides now. Great. Yeah, thank you again, Jasmine, and thank you, Anushri and Stuti, for the kind invitation. I'd love to be with you in person, but this virtual online format opens up a lot of possibilities for communication around the world. So today I'm going to tell you a bit about a number of projects that we've been working on in recent years. If I can just get my slides to advance. There we go. Hopefully you can see that second slide now. So I'll be talking about a number of disorders that we've been modeling and how genes and environment interact, as well as the role of epigenetics. So I'll talk a little about a mouse model of Huntington's disease. I'll speak briefly about mouse models of schizophrenia, as well as models that are relevant to depression and anxiety disorders. I'll also be talking about various environmental factors, including the role of mental and physical activity, the role of stress, and I'll mention the role of diet as well. So to start with, I want to introduce Huntington's disease. So there's been a number of revolutions in science in recent years, and one of them has been genetics, which is going to power the 21st century specialties of precision medicine and personalized medicine based on genomics and other technologies. So it's not commonly known that approximately half of the human genome is repetitive DNA, or what I call repeatome. Within that half, there's over 1 million so-called tandem repeats. And one of these tandem repeats causes Huntington's disease. The many other tandem repeats have largely unknown functions, and there's at least 50 other disorders that are monogenic disorders caused by different tandem repeat expansions. But then there's new evidence, including this year published in Nature, that disorders like autism may have major contributions from tandem repeat mutations. And so this is probably just the tip of the iceberg in terms of how this repeatome is involved in a whole range of different human diseases. But focusing on this group of so-called monogenic tandem repeat disorders, you can see examples of them here. And Huntington's disease is the most common of at least 9 so-called polyglutamine diseases, because the tandem repeat, or CAG, this genetic stutter that encodes an expanded tract of glutamines, is found to cause Huntington's disease. So in that sense, it's quite extraordinary. Hopefully those listening aren't from a Huntington's family. So if you're not from a Huntington's family, your Huntington gene has between about 5 and 35 of these CAG repeats, encoding between about 5 and 35 glutamines in the Huntington protein. Someone who will suffer and die horribly of Huntington's often just has a handful of extra repeats. And keep in mind that the genome's 3 billion base pairs of DNA. So the difference between life and death in this type of genetic disorder is extremely fine indeed. In terms of trying to understand Huntington's, we've been trying to understand how the expanded polyglutamine can induce a toxic gain of function in a whole range of cells, particularly neurons in parts of the brain, such as the cortex and striatum, but also exploring other ideas, such as how it might change the normal function of this Huntington protein. This gene was first described by George Huntington in 1872. Not the gene, the disease was first described by George Huntington as a physician. It wasn't until 1993 that the gene causing Huntington was discovered, the gene mutation. And so George Huntington, as a physician, only ever published one paper. So according to today's metrics, he'd have a H-index of 1, which wouldn't be that impressive, but he really started the Huntington's field by identifying this as a disorder that was being passed down in families. If a parent had Huntington's, approximately 50% of their children would inherit the mutation and get the disease. And so it's a very sobering fact that almost 150 years after this disease was first described, we still have no disease-modifying treatments. We have no way to cure it. These people are still dying of this horrible disease, even though in theory, being caused by a single gene, it's meant to be quite simple. And it's been over 25 years since the gene was discovered in 1993 as well, and we still don't have an effective treatment or cure. So this repeat is expressed throughout the brain and throughout the body, as I'll mention later, but particularly affects populations of neurons in the brain, including the cerebral cortex and the striatum, a part of the basal ganglia. So for each of these disorders, we can put together a schema, and here you can see this genetic stutter, the CAG repeat stutter, the tandem repeat, that in Huntington's patients expands, and therefore they have an expansion of this polyglutamine tract of amino acids in the Huntington protein. And beyond that, there's a cascade of molecular and cellular changes, which ultimately give rise to the symptoms of Huntington's disease, which are a triad of symptoms, including cognitive deficits that culminate in dementia, psychiatric symptoms, the most common of which is depression, and the movement disorder, including chorea, these writhing dance-like movements that are typical of Huntington's and distinguish it from other movement disorders, such as Parkinson's disease, which has a typical tremor and so on. There's various changes at the cellular level which we think are involved, and we've proposed this idea of pathological plasticity. Forms of cellular plasticity, such as adult neurogenesis and synaptic plasticity, which are involved in various cognitive and affective functions, may be disrupted in Huntington's, and this may give rise to particular cognitive and psychiatric symptoms. All right, so in terms of trying to understand disease, what we need is a preclinical model. And one of the advantages of an autosomal dominant monogenic disorder like Huntington's disease is that you can take the gene mutation and you can then insert that within the genome of essentially any other species, laboratory species. And this has been done widely. In fact, it has been inserted into the genome of sheep and pigs. There is no goat model yet, but we, like many others, have chosen for many reasons to focus on mice and transgenic mice, which have a lot of utility and power. But they also have genomes very closely related to humans, and their brains have all of the structures that you want to see as a related mammalian species that we can model the human brain and in fact other organs. So here's the main mouse model that we've used. This transgenic mouse model of Huntington's expresses a human Huntington's gene fragment, and therefore these mice, just like a Huntington's family, where one of the parents will have the gene mutation and get the disease and pass it on to half the kids. We breed from a transgenic male Huntington's mouse, bred with a female perfectly genetically strain matched females. And therefore half of the offspring mice have the Huntington's gene mutation, and the other half lack that mutation, but are otherwise genetically identical. So perfect genetic control, which you can't get in a human study. So there's a lot of power here. And we always randomize these offspring mice into different experimental groups and do the experiments blinded so that we have very high quality data and a lot of control. And so over many years, in fact, a couple of decades now, we've been able to show not only do the affective or depression-like, the cognitive deficits modeling dementia, and the motor deficits modeling chorea and other symptoms, not only are these seen in the mice, but changes in the brains, looking here in coronal section through these stained sections, brain sections from mice. You can see on the left the Huntington's and on the right the wild type. And the Huntington's over time, just like a human Huntington's disease, this is a striatum, and as it degenerates, this ventricle becomes larger because this tissue is degenerating, and the cerebral cortex is also degenerating. So it very closely models human Huntington's disease. We can generate a timeline because this is a progressive neurodegenerative disorder. So we and others have been able to show over many years that there's an onset of these affective or depression-like signs that occurs very early, and the cognitive deficits modeling dementia occur very early, and they precede the motor deficits modeling chorea and other movement disorder. And things like neuronal cell death are very late. So this closely matches human Huntington's disease. And what we want to work out are the earliest molecular and cellular changes involved in the disease process. And a subset of these molecules are going to be targets for new therapeutics, including drugs. So if we can understand what the key molecules are, some of those can be targeted with drugs. And that's how we will eventually be able to prevent onset or delay onset and treat those with symptoms and ultimately cure this disease. That's the ultimate goal. We've been exploring this in a whole range of ways. And one of the ways which Huntington's and some of these other tandem repeat disorders are disrupted is epigenetic dysregulation. At this point, I should explain to this audience what epigenetics is. So you can think about epigenetics in two main ways. One way to think about epigenetics, if you're bookish in interest, is to think about the cells in the body each have a genome, a copy of the genome, and think about that genome as having about 20,000 genes. Think about each of those genes being a word. And so that's a little handbook, a guidebook to create a human body and have it function. And so if you think about the letters of DNA making up these words in the genes, then think about epigenetics being underlining, bold, italics, or even fluorescent markers that dictate the extent to which each gene is turned on or off. The other analogy I use for epigenetics is a musical one. Think about the 20,000 genes as 20,000 musical instruments. Then think about epigenetics being the musicians. So when you put the musical instruments and the musicians together, you get the symphony of life. All right, so this is just one aspect in Huntington's. Disrupted epigenetics means there's disruption of gene expression in particular cells within the brain, and we think in other parts of the body as well. We think one part of the brain particularly affected are these synapses, keeping in mind the human brain has about 100 billion neurons and trillions of synapses, these connections between them. Each synapse itself has a lot of complexity. So you can see all these molecules, mainly proteins, making up the synapse. And over the years, my group and others have shown that particular genes and their encoded proteins are disrupted in Huntington's. And this disrupts neurotransmission at particular synapses, including synapses that use a neurotransmitter glutamine, or GABA, acetylcholine, serotonin, also dopamine, for example, and even the neurotrophin BDNF. And you can see on the other side the receptors and potential downstream consequences of disrupting synapses. It's so-called synaptopathy, we call it. All right, so moving now on to the core message in this talk, which is about genes and environment and how they interact. And it's leading into this key experiment that we did now exactly 2 decades ago, or published it 2 decades ago. And this was the first evidence in any genetic model of a human disease that environmental stimulation could be therapeutic. The approach we were to take was environmental enrichment, which increases cognitive stimulation and physical activity. We later, as I mentioned, looked at stress and other factors. Since we first demonstrated this in the Huntington's disease mice, our colleagues around the world have followed this up in rodent models of Alzheimer's disease and Parkinson's and so on, and found related effects. So here's environmental enrichment in mice. Relative to standard housing conditions, the mice have increased novelty and complexity in their environment. This increases levels of sensory stimulation, cognitive stimulation, and opportunities for voluntary physical activity. So in this initial experiment, at weaning, which was around 4 weeks postnatal in mice, when they're weaned away from their mothers, the mice, which are, as I mentioned before, Huntington's and strain-matched wild-type control mice, were randomized into 2 environmental conditions, either standard conditions where they have soft bedding and unlimited food and water and other mice of the same sex, or environmental enrichment where they had this increased cognitive stimulation, physical activity. And so these were our original findings. And this was done with my first PhD student, Anton van Dellen. And we were really struck by these findings. And what we found, you can see along the bottom, this is a test of motor coordination. It's called a horizontal rod test. It's a test which is sensitive to the movement disorder that develops in these mice and models chorea and other motor deficits in HD. You can see the wild-type control mice of both groups never show this motor deficit as expected, because we don't expect them. They don't have the Huntington's gene. They don't develop a movement disorder. Those under standard housing, the more boring with less stimulation, develop by 5 months of age. 100% of them up here develop this movement disorder, motor deficits. And this nicely models the 95% of Huntington's patients who develop adult onset symptoms. So it's a very accurate model. But strikingly down here, you can see that the environmentally enriched Huntington's mice showed a dramatic delay in onset of Huntington's disease. Now, this is a very important finding because at the time we published this, Huntington's was described as the epitome of genetic determinism. In textbooks and elsewhere, it was described as 100% genetic. And therefore, it was a major breakthrough at the time. Importantly, this is a very robust and reproducible finding. Not only have we reproduced it many, many times, but our colleagues in independent preclinical models of Huntington's disease, in independent labs, have been able to replicate this key finding. And in fact, showing that the effect size is bigger than just about any other intervention in Huntington's mice, including a whole range of different drugs that have been used in these mice. Importantly, like many of our discoveries in these Huntington's mice, it has translated to the clinic. So I remember when I first presented the data in this Nature paper in Boston, when I was traveling there for a meeting, And I think it was just before it was published. And so in the audience was Nancy Wexler. And Nancy's a dear friend and a leader in the Huntington's field. It's a very famous family. Her mother died of Huntington's disease. Her father, Milton, based on this, was a driver in raising a lot of money for Huntington's research over a long period of time. And obviously, Nancy and her sister Alice, who's also famous for having written a number of books about Huntington's disease, were obviously 50% at risk. And having watched Nancy as a friend and colleague over all these decades, we always knew that she was 50% at risk, but obviously many people in these families don't disclose their genetic status, or they may even choose not to take the test. But more recently, we have seen Nancy develop these dreadful Huntington symptoms. It's very clear she's very symptomatic and she's clearly dying of Huntington's, and we can't develop a treatment. So to have a friend like her develop this horrible disease brings home the importance of this medical research. And I think for everyone in this audience, for those who aren't sure what their future holds or their career, I'd say to you in this audience, firstly, that doing neuroscience in the early 21st century is like doing physics was in the early 20th century. Neuroscience is the great frontier, both in terms of basic science, but also in terms of medical research. All the greatest burden of disease and the greatest growing burden of disease is brain and mind disorders, including a whole range of neurological and psychiatric disorders. So I don't think you could really find a better career. And even for those who are looking for some security, perhaps, I often tell the story to students about Steve Jobs, who founded Apple. And as you may know, he was worth over $30 billion when he died. He died of cancer. And so Steve Jobs, despite being worth over $30 billion, could not stop himself dying at 56 of cancer. So what does that tell you in a world where although there's certainly a lot of problems, there's a growing wealthy and middle class, a lot of typical working people have more disposable income, and what everyone wants is to live longer and live healthier. It's a commonality amongst everyone. The only way that we can help everyone live longer and live healthier is health and medical research. And so on first principles, it could be argued that science and health and medical research is probably the most secure career of the 21st century. But getting back to this evidence, together with Nancy, they did this kindred study and were able to translate our work and show in this large kindred in Venezuela, this was the founder effect, where a Spaniard traveled to Lake Maracaibo in Venezuela hundreds of years ago. And he carried with him the Huntington's mutation in his genome, and he passed it on. He had a very large number of kids and grandkids and so on, and created this enormous founder effect where 1 in 4 of these people in Lake Maracaibo in Venezuela had the Huntington's gene and were going to die of Huntington. So horrible for them, but very powerful for this cohort of North American clinicians and geneticists who studied them. And that's how they found the gene with these Venezuelans. And so Nancy and her team went back and was able to show for a given CAG repeat length. So when that stutters longer, you get the disease onset earlier. However, there's a whole lot of variance that's not fully explained by the CAG stutter length. And of that variance, it can obviously be genetic and or environmental. And the kindred analysis showed it was more environmental than genetic, thus translating our discovery in the mice. And also colleagues led by Martin Delaticchi in Melbourne did retrospective epidemiology and provided additional evidence that these environmental factors are at play in clinical Huntington's disease. So then we had this idea of molecular and cellular mediators and environmental modulators of Huntington's disease, and we're trying to build this into our model. We looked at a whole range of different changes caused by Huntington's disease, including gene expression and epigenetic regulation that I mentioned before, but also the idea that environmental enrichment can have effects on gene expression in particular brain cells. And then these forms of cellular plasticity, adult neurogenesis and synaptic plasticity, we and others have discovered that these are affected by Huntington's and may give rise to the cognitive and maybe the psychiatric symptoms as well. And environmental enrichment can enhance cellular plasticity, can enhance adult neurogenesis and synaptic plasticity. So we've looked at a whole range of different technical approaches in the brain tissues of these mice, including transcriptomics, epigenomics, proteomics, and metabolomics more recently in these key brain regions. And some of it has brought us back to the synapse here. Again, a different cartoon of the synapse with glutamate released onto these glutamate receptors. And our other proteins we think are heavily involved in these forms of synaptic plasticity called LTP and LTD, which we think are intrinsic for learning and memory. And therefore, when it goes wrong, disrupting learning and memory can cause dementia. And so there's particular proteins at the synapse that we've shown have disrupted in Huntington's disease, and a subset of them are rescued by environmental enrichment. And we use this to identify new targets for potential therapeutics, not just for Huntington's, but potentially other disorders like dementia, depression, and so on. One thing we have been looking at is this form of cellular plasticity called adult neurogenesis. It occurs in pockets of the adult brain, including the dentate gyrus of the hippocampus. And we and others have been able to show that the Huntington's mutation disrupts adult neurogenesis in the hippocampus. And also in some of these preclinical models, there's correlates of decreased adult neurogenesis that correlate with depression and cognitive deficits, modeling depression and dementia in humans. And these various factors here are disrupted in the Huntington's mice. We and others have shown that. On the other side, in preclinical models with increased adult neurogenesis, there's correlations with antidepressant-like effects and cognitive enhancement. And these factors include environmental enrichment, exercise on its own, and these SSRI antidepressant drugs like Prozac or Zoloft. Okay, so the next challenge was, how do you model something as complex and human as depression in an animal? And it's not easy, and we're as self-critical as the next. But one advantage we have in trying to use preclinical models of psychiatric disorders is that there are some drugs that are at least partially effective in human populations, like antidepressants for depression, or anxiolytics for anxiety, or antipsychotics for psychosis. And so we've taken on this challenge of trying to model depression, and we made the discovery. We know that up to 50% of individuals with the Huntington's gene develop depression. And we're able to show that the expression of this gene in Huntington's can generate depression-like behaviors, and that these behaviors respond to clinically effective antidepressants like sertraline, which is marketed as Zoloft. And so we have a very accurate model of depression within this transgenic mouse model of Huntington's disease. We've also been able to show that environmental enrichment has antidepressant-like effects in these mice. And we know in the human population that exercise can have antidepressant-like effects in the general population with depression. So we have explored exercise as one of these components that environmental enrichment induces. And we've been able to show that voluntary wheel running in mice not only ameliorates an abnormal stress response in these mice, but ameliorates the cognitive deficits modeling dementia, the affective changes modeling depression, and also the movement disorder modeling chorea. This has taken us into a study of stress responses, including this hypothalamic pituitary adrenal axis, this HPA axis, which in humans, the stress hormone is cortisol. In mice and rats, it's called corticosterone, but it's the equivalent stress hormone. And this discovery we made involved the idea that the HPA axis is disrupted in these female Huntington's mice. I didn't mention before, but the female Huntington's mice are the ones that show the clear depression-like behaviors. And in fact, in clinical depression, it's twice as common in women as men. So the sexual dimorphism in the mice closely matches human sexual dimorphism. So this is important, that the HPA axis function is disrupted. We were able to show that this could be corrected by environmental enrichment, and cultured cells from the adrenal gland would still release too much stress hormone in vitro, because the Huntington's gene is expressed even in the adrenal glands, even in vitro. But this could be corrected by environmental enrichment in vivo, the idea being that some epigenetic memory of the experience, environmental enrichment in vivo, is carried into vitro in the dish, these adrenal cells, where they have an epigenetic memory of their experience in vivo, and that corrects their abnormal release of this stress hormone, CORT. We've also looked at other models of stress, including giving chronic stress hormone in the drinking water, which elevates levels of stress hormone. And we're able to show that this accelerates onset of a cognitive deficit, modeling dementia and Huntington's. And physical stress too can accelerate specific symptom onset in these mice. So stress is a highly relevant environmental factor, and our clinical colleagues are following this up in their clinical studies now. So we've used this to come up with I've proposed a concept called environmemetics. So identifying key molecules involved in the response to these environmental modulators could allow us to use these molecules as therapeutic targets for these environmemetics, which would mimic and or enhance the beneficial effects of cognitive stimulation and physical activity. So what might an environmemetic look like? Well, we've trialed a candidate environmemetic, and it's called N-acetylcysteine, or NAC, and we've shown that it's very much beneficial in these Huntington's mice, partly by being an antioxidant and having beneficial effects on mitochondria, but partly also by modulating the glutamatergic system. And so this was showing that this NAC could also have antidepressant-like effects, which I mentioned before, that environmental enrichment can have antidepressant-like effects. And so we've shown various molecules, again, at this synapse that has glutamate onto glutamate receptors. And not only shown its benefit in the mice, but now we have some funding to do a clinical trial with clinical colleagues in Australia, where we're going to see whether N-acetylcysteine clinically can delay onset of Huntington's. It's the first trial of its type, not just to treat symptomatic patients, but to see whether we can delay onset of Huntington's using this drug that had such strong effects in the mice. So I mentioned that the genes expressed, the Huntington gene expressed in the brain, but also the body. And that's the same in our transgenic mice. And we and others have shown over a number of years that Huntington's is not just a brain disorder. It's a disorder of the brain and the body. Now, what we looked at next was wanting to look at the gut and looking at these microbiota. So a fascinating revolution again in biomedical research in recent years has been a study of the microbiota and the microbiome, the molecules that they create, including the genomes within these bacteria. So the interesting thing is we have about 1 trillion cells in our bodies as humans, and we have more than 1 trillion bacteria in our gut. And so if you just count the number of cells, all of us are actually more bacterial than we are human based on that alone. So it's a very complex system, and there's bidirectional communication between this microbiota in the gut and the brain. So we wanted to study this in the mice, and it's quite a simple experiment. You collect fecal samples from the mice, and then Geraldine Kong isolated the DNA and did this DNA sequencing that allowed these bacteria to be characterized and into the different classes of bacteria. And then she was able to show that the Huntington's mice had altered gut microbiota. We've again translated this with clinical colleagues in Melbourne and showed in clinical Huntington's patients, and even those who are gene positive and pre-symptomatic, there are changes in the gut microbiota. And that's something that we're currently following up, which has potential major therapeutic implications. This is a new paper where we've gone even further with so-called shotgun sequencing, where we can get right down to individual species of bacteria and able to link that to the blood and these metabolites present in the blood, some of which are released by the bacteria and some of which signal to the brain. And so we're pursuing this to see how we can link the gut to the brain and maybe develop therapies to treat brain symptoms via the gut. This is an overarching model which links Huntington's to Alzheimer's, to Parkinson's, to motor neuron disease, and the idea that the gut microbiota are disrupted in all of these diseases, but also that environmental factors could act on the brain via the gut, and that's something we're actively exploring as well. I'm moving now on to another project which involves gene-environment interactions in schizophrenia. Schizophrenia is a far more complex genetically it's far more complex than Huntington's disease, for example. So we need to be thinking of the genomes, but also the epigenomes, the epigenetics from conception through to old age. But we think that the environment might start with your parents and grandparents. And I'll mention later on this epigenetic inheritance of acquired traits, suggesting the environment starts very early indeed. With schizophrenia, we're dealing with very complex cognitive deficits and psychotic symptoms, including negative or affective symptoms. And it's a very complex and devastating disorder. We've been looking at one pathway in particular and looking at this metabotropic glutamate receptor 5 and its signal onto a protein called PLC beta 1. I won't go into details here, but suffice to say there's evidence that this metabotropic glutamate receptor and muscarinic acetylcholine receptors are involved in this particular signaling pathway in the cerebral cortex. And following up on this work, we were able to look at the behaviors of these mice and look at endophenotypes which model clinical schizophrenia. And what we found was that these endophenotypes in the mice that are used to model schizophrenia were altered. And so this says that this is an accurate model of schizophrenia in terms of modeling symptoms. And even given an antipsychotic drug, clozapine, we could change these behaviors in the mice. And so it suggests that this is an excellent mouse model of schizophrenia. That was under standard conditions. And so we repeated this under these environmentally enriched conditions. And what you see here is that importantly, and when we think of standard housing as really a sensory motor deprivation state as the negative environmental condition, it's only when you combine that negative environmental condition, standard housing, with the knockout mouse, we consider that a form of genetic predisposition, so negative in genetic factors. And when you have negative genetic factors and negative environmental factors, you get here this behavioral endophenotype that models schizophrenia. And that's exactly what we're trying to model in these kind of psychiatric disorders. We're also able to show that a different mouse, which has a gene mutation in this metabotropic glutamate receptor 5, also models schizophrenia very closely. And that's just showing changes in the cerebral cortex. And the cognitive deficits in schizophrenia are very poorly treated. They're quite devastating, and antipsychotic drugs tend not to correct them at all. And antipsychotic drugs have major side effects. So it's not a well-treated disorder at all. However, environmental enrichment corrects these cognitive deficits in this model. So if we can work out how environmental enrichment does that, we could develop new therapeutics that correct the cognitive deficits in schizophrenia, which is a huge unmet need. This is showing here, I'll just go on to this. You can see here, this mouse, there's an iPad or tablet device at the front. It touches its nose and gets a strawberry milk reward at the left, if you're looking on the left side here. So it got the answer correct. Now look on the right side here, again, the iPad or tablet device up the front. These are automated cognitive touchscreen testing chambers. It touches its nose, but the light comes on. So the mouse knows it got it wrong and it doesn't come to the back to get its strawberry milk reward. These closely model neuropsychology touchscreen tests in humans, they're totally translatable. We've been able to show that this mouse model of schizophrenia has cognitive deficits that model clinical schizophrenia on these translatable tasks, and that environmental enrichment can enhance cognition on these translatable cognitive tasks. Trying to put this together, we think going back to Conrad Waddington in the 1940s, who coined the term epigenetic landscape, we think part of what's going wrong in disorders like schizophrenia and autism is this disruption of the epigenetic landscape. So the proposal here that epigenetics means that there's expectant brain development, expects certain environmental factors, and the brain development's normally canalized through evolution to have a very reliable developmental path. However, decanalization or disruption can occur in the absence of expected instructions like the right environmental stimuli. Decanalization can also occur when there are unexpected instructions like major stressors during pregnancy for the fetus, or major stresses during adolescence, which are very relevant to schizophrenia. linking this epigenetics, we want to translate this by thinking about these environmental stimuli on particular cells in the body having particular epigenetic effects. And once we can understand that, these little yellow and green circles refer to therapeutics, which may include drugs, which would target particular epigenetic molecules, so that these might be called epimimetics, which mimic the beneficial effects of specific epigenetic modifications. Now, furthering this epigenetic approach, we've been also looking at how environmental factors not only affect the individual, but can affect the next generation and maybe grandchildren as well, not just through mothers, but also through fathers, the paternal line. And we and others around the world have been actively exploring this question, which has almost Lamarckian connotations. You can see here the first experiment we did where, again, in the drinking water, we increased stress hormone, or these cortisol levels, in half the mice, and the other half were just given water. They were the control group. And this was done for 4 weeks. And then we mated the mice with control mice and produced offspring mice. And so in the F1 offspring, if the fathers had increased stress hormone modeling stress, the offspring, and the male offspring in particular, had increased anxiety. We obviously then proposed that this could be the epigenetics of the sperm that is altered and passing on this information, and explored this. And I'll just focus here on these small non-coding RNAs present in sperm. Using RNA sequencing technology, we were able to show that specific microRNAs were altered by the increased stress hormone in the males. This could be linked to specific mRNAs, these transcripts in the brains of the offspring, suggesting potential epigenetic mechanism for this intergenerational epigenetic inheritance of an acquired trait. Then just giving exercise alone to the fathers for this 4 weeks before conception also had behavioral effects on the offspring that had cognitive and affective components. Again, we looked at these small RNA molecules in the sperm of the mice and found many of them were changed. And also we looked with this environmental enrichment in the fathers, and that was found to have effects that traveled right through to the F2 or the grand offspring. So putting all this together, the idea that these factors that a male might be exposed to before conception might change the sperm epigenetics, and part of this might be through circulating factors in the bloodstream based on our colleagues' recent paper. There's obviously various environmental factors transmitted through the mother as well. I haven't talked about those today, but the combination of the genomes and epigenomes of the father and the mother can lead to changes in the offspring and what I call intergenerational epigenopathy, that is epigenetic contributions to disease being passed through generations. And in some cases, we might think about that some positive environmental factors could build in epigenetic resilience. And this is a new concept which I've recently published. We've also been thinking about these environmental factors like stress, environmental enrichment, and physical activity, modulating the sperm epigenome, and how this might change things like brain development, maturation, and function, and ultimately how that might relate to changes in the offspring. More recently, we've also looked at other environmental factors, including infection in the males before conception, and with colleagues in Melbourne have shown that this can change the offspring. That's something that we're actively exploring. One other factor that can affect the offspring via males and their experience before conception is diet. And this is something we're also currently exploring with Karina Bodden and Amy Reichelt, the effect of junk food or Western style diet fed to fathers before conception can have changes in the offspring through the sperm. So obviously all of these factors like diet and stress and physical activity, which are changing radically in modern populations, particularly Western populations, but I know in countries like India and other countries, lifestyles have changed a lot in recent decades as well. And so the scary thing is here, from a public health perspective, we could be setting up epidemics in the next generation through both the paternal and the paternal lines, this so-called epigenopathy. So what we need to do is find ways to prevent this epigenopathy transmitting to the next generation if possible. If it does, we need to diagnose epigenopathy and have tailored approaches like epimimetics, where we can have precision medicine and personalized medicine approaches to it. So finally, I used to say we're all dealt a genetic deck of cards at conception that we can do nothing about. That's true, but we're also apparently dealt an epigenetic deck of cards at conception. The difference being that we may be able to do something about that because some epigenetic modifications can be reversed. You can see down the right side here, these red arrows refer to brain development that's starting to become dysfunctional. We know that positive environmental factors like mental and physical activity might build in neuroprotection via the so-called brain and cognitive reserve. Even a brain that's become dysfunctional during development may still be able to undergo functional compensation. And even someone who's developed a brain disorder may be able to have new therapies such as this environmemetics or epimimetics. We hope we'll be able to push the brain over to this side of healthy brain maturation and aging, which is where we all want to be. So finally, I'd like to acknowledge these wonderful people, these students and postdocs and other staff who've done all of this fantastic work, and bold are ones who particularly contributed to data I've shown today. They're really an inspiration to me. They're just wonderful people, particularly in this COVID-19 pandemic year, which I know has been hard for many of you, but it's been particularly hard also in Melbourne that was affected, and the determination and the dedication of these people, I can never thank them enough. So I'm happy to take questions now, and I thank you all for your attention. Thank you.
Thank you so much, Professor, for a lovely conversation and the entire presentation. And I honestly loved the part where you explained epigenetics with the music analogy. And gladly, the audience also loved that bit a lot, as I saw from the messages that we got. And yeah, we would be taking up the questions right now. So the first question that we have is...
Happy to take them, thank you.
Yeah, yes, sir. Sorry. So the first question that we have is from... okay, it's from Jyotirame. Could it just be that the gut microbiota change and the disease state are correlated without having any causative mediatory effect, as in they're both the effect of environmental change?
Look, that is a great question. We're also always thinking about correlation versus causation. That's a great question. And so we're doing experiments now to test that. And so as you can imagine, there are approaches whereby you can test that. There are approaches like fecal matter transplant, for example, is one approach where you can manipulate the microbiota in the gut and see whether the brain changes and behavior changes and so on. So these are testable hypotheses which we are changing or testing. And another aspect is that Huntington's has symptoms outside the brain, so including gastrointestinal symptoms. So it's possible also that the microbiota changes primarily cause the gastrointestinal symptoms, but we're open to the possibility that they contribute or modulate to the brain symptoms, and we're testing that now. And that's the advantage with mice. You can test all these hypotheses and address mechanism and distinguish causation from correlation.
All right, so also another question that we have from Bandhan Goyal is that they understood the analogy, but they were a bit unclear about epigenetics from the psychological perspective.
So the question is, what about epigenetics from a psychological perspective?
Yes, sir.
Yeah, so epigenetics, it's a very broad concept. And of course, development of a human involves epigenetics. So in order for cells to have different fates during development and create a human being, epigenetics is involved in all of that. But even in an adult, with psychology, we think of it more as cognitive neuroscience, you know, how the brain generates thoughts and emotions and so on. We think of it from that perspective. And so epigenetics has been heavily implicated in cognitive processes like learning and memory. My colleagues in Australia are working on this. We're collaborating with them to work on this. And so, you know, experiences that induce learning and memory, it appears that one of the ways in which learning and memory is laid down in the long term is through epigenetics. Epigenetic modifications can lead to neurons and synapses changing their function in the long term. And so cognitive functions like learning and memory seem to rely very much on epigenetics.
Yeah, so even I understood that bit now. Also, there is another question from Anushri Krishnamurthy, that is there any gradient in the CAG that repeats that decides the severity of the disease? And if so, how is this quantified?
So a gradient of disease, the severity, is that the question? How do you quantify the severity of Huntington's or other disease?
It's specifically related to the CAG that repeats the severity of the disease. I'm not sure which disease was mentioned. She's talking about Huntington's only.
Right. Sorry, your voice was breaking up, so I may not have got the question correctly, but I'll, in terms of, we have very accurate quantitative tests that accurately measure the extent of a cognitive deficit, the extent of the depression-like behaviors, the extent of the movement disorder. So we always do quantitative analysis, which gives us quite a good range. So we can detect the mice where the Huntington's are normal and no different from the wild type littermates, and then they subtly start to become different. And then we can look at late disease where they're very different. So our tests are very quantitative, and we can map onset of each endophenotype or cognitive affective motor change over time, which gives us very accurate quantitative measures. And our clinical colleagues have accurate quantitative measures as well.
Okay, sir. And this is something that I wanted to ask myself as well. If environment plays a crucial role in the onset of the disease, then can't environment itself be used to gradually deplete the harm done, even if it's like at the initial stages?
Yes, well, that's what this clinical translation for Huntington's, and we think other disorders, is that we think environmental factors need to be trialed in all these disorders, and they are, based on our work, they're being trialed for Huntington's. However, the environmental factors alone, something like Huntington's, can't stop the disease dead in its track. They can delay onset, they can slow progression. However, on top of that, we need new therapeutics like drugs, and they may work synergistically with things like cognitive stimulation, physical activity. And you have to keep in mind that as someone develops dementia or movement disorder, they become less and less capable of doing physical activity or even engaging in cognitive stimulation. So as they become more symptomatic, it is harder to implement these kind of environmental interventions as well.
All right, sir. And the next question we have is from Shresh. He says that what are the possible neurobiological mechanisms stimulating adult neurogenesis due to environmental enrichment?
Yeah, great question. So this has been fairly well studied, and so the traditional view was that the physical activity could increase the birth of cells in this dentate gyrus of the hippocampus, and that the cognitive stimulation might mean that more of these adult-born neurons survive and form synapses and integrate with the circuitry. So at the level of molecules, there's key molecules like this brain-derived neurotrophic factor, or BDNF, that we and others have implicated in this process. But there are other molecules like serotonin that have been implicated. There are, even at the cellular level, as well as these adult-born neurons, there are other glial cells like microglia and astrocytes that have been implicated in the process as well. So it's quite complex, but we think one of the molecules that links, for example, physical activity to these changes in cellular plasticity could be BDNF, for example.
Okay, sir. And Meghna wants to ask if the use of probiotics would have any effect.
Probiotics?
Yes, sir. Probiotics.
Yeah, look, that's a fact. We are pursuing this as well. For those who don't know, probiotics refer to the delivery of live bacteria of particular species to the gastrointestinal tract. And that's one of the reasons that we did the metagenomics, where we identified all of the bacterial species altered, which will give us an ability to go after specific probiotics. However, an approach that we're also interested in is prebiotics, which are more related to dietary components, like different levels of fiber and other dietary components, which is what the bacteria eat in the gut. And so prebiotics are another way we're looking at testing this. We have been involved in a probiotic clinical trial with the Huntington's patients, which is a manuscript under review at the moment. But that will require a lot more work to get the right probiotic and get the right intervention. It's very early days in terms of translation.
All right, sir. Yes. So, Jyotirame also wanted to ask, the gut microbiota change and the disease state are correlated without having any positive mediatory effect, as in they're both the effects of environmental change.
Yeah, look, we haven't yet got evidence, and we're not claiming it, that the gut microbiota changes are causing the brain changes. They may be correlated, and both from the perspective of the Huntington's gene, maybe independently changing the brain function and changing the gut microbiota, because it is expressed in the gut as well, the gene. And then the environmental factors may be independently affecting the gut and the gut microbiota and the brain. But we are doing these functional experiments that will distinguish correlation from causation. That's what we can do in a mouse model. It's impossible. Most of these human studies, you simply can't demonstrate causation. And that's why you need good preclinical models like mouse models to go after mechanism causation.
Okay, sir. And there is a question from Ruth as well, that if epigenetics can be combined with low dose ionization therapy for the treatment of HD.
Low dose ionization. Yeah, sorry, I don't know anything about that approach. Low dose yeah, I'm not familiar with low dose ionization at all. But what I would say, and particularly because there's, high school students and so on in the audience, if you want to be a scientist, if you are already training to be a scientist, you must be skeptical and critical. One of the problems you face, and we all face, is information overload and distraction. The internet, social media, there's just so much information, so much distraction, and so much fake news. And to be honest, fake science, really, where people are claiming something scientific when it's not. And that's why we need quality peer-reviewed journals where scientists test hypotheses, they publish work, and their peers critically evaluate it. And just because it's published in a peer-reviewed journal doesn't mean it's the truth. But there's different levels of evidence. Same with clinical trials. There's low-level clinical evidence. And we've seen this with COVID-19, including President Trump was making claims that had no science, no evidence, but he was stating it around to an international audience when there was no scientific evidence at that point. Whereas the medics, including Dr. Fauci, were saying, no, you need proper clinical trials, and the vaccines now are gone through phase 3. That's high level evidence. Whereas people were claiming treatments based on no science or extremely low level evidence. And therefore, if there is a new, potential treatment like ionization, you need to be critical and skeptical of it, and you need to test it rigorously. And, that's why we're looking at mechanisms. If there's no potential mechanism, then why do you think something like that would work? That's part of learning to be a scientist.
Wow. I mean, yes, sir, I definitely agree now that in times like these, we need to actually put in a very big effort to understand what is possibly right and could or could not work. Which brings me to the next question, that is from Hira Adik, that was the significance tested based on episodes of stress, like increasing stress at some point and decrease or null stress at other points? How is that going to affect the epigenetic?
Well, great question. And we haven't, that's an area we're wanting to pursue more, is, different types of stress, what effect they have. But we know that stress can have big epigenetic effects. A typical example is post-traumatic stress disorder. So the idea, again, using preclinical models, these major stressors seem to have major behavioral and cognitive and affective changes, partly through epigenetics. They leave a kind of epigenetic memory in parts of the brain, like the prefrontal cortex, that means that someone has, even though it was an acute traumatic event, they've still got effectively an anxiety psychiatric disorder years later. And that's one way, something like PTSD can have such long-term effects is via epigenetics, we think.
Okay, sir. And I think Pranjal also wanted to ask if you could tell us a bit more about the clinical trial that you mentioned.
Yeah, so the clinical trial for N-acetylcysteine, there were a couple I mentioned, but the one for N-acetylcysteine is with clinical colleagues. They include neurologists, psychiatrists, clinical neuropsychologists, and it's really taking the compound, and it's going to be a long trial because what the unique aspect is to target people who have the gene mutation. They don't have the disease symptoms yet, but based on modeling, we think they're due to get the symptoms in the next few years. And in that type of trial, you can, if you said this patient was expected to get the symptom in 2 or 3 years, then after 3 years, if they haven't got the symptoms, you can start to draw conclusions that the drug compared to the placebo was effective. And this has direct parallels with Alzheimer's, the idea that there's a prodromal phase, like mild cognitive impairment before Alzheimer's as a major form of dementia. And so we're kind of pioneering this approach that could then be taken into things like mild cognitive impairment and trying to stop someone with mild cognitive impairment developing Alzheimer's or other forms of dementia.
All right, sir. And I think this brings me towards the next question that we have from Asta, that is it at all possible to develop a medicine for treating patients who have already developed symptoms of the Huntington's disease?
Yeah, so to stop them developing symptoms? Is that the question?
Sorry, sir. Could you just repeat the last thing that you said?
So was the question about developing something that stops the development of symptoms?
It's for treating the patients who have already developed the symptoms. I think.
Right. Yeah, look, there are current clinical trials which are focused on trying to treat people with symptoms to slow progression, really. Reversing symptoms is a lot to ask. And so there are various clinical trials trying to, slow progression. Whether they'll be able to reverse symptoms, that's a big ask. I think with most disorders, the earlier the better. The old phrase was an ounce of prevention is worth a pound of treatment or whatever. I can't remember the exact phrase, but the idea is if you can get in early and do something, and prevention's much more powerful than treatment, and the health economics is much more powerful as well. So and that's what COVID-19 pandemics taught us. We need to, how do we prevent the next pandemic? How do we, if we have another pandemic, how do we get in really early and nip it in the bud? I think the world has to learn from this pandemic and has to take the approach in terms of public health to other disorders, not just a specific virus. And you might argue more broadly that if we can change human behavior so quickly within a few months, why can't we do that for climate change? Why can't we change human behavior to stop climate change? If we can change human behavior in a few months to try and stop a virus, it raises many questions around how we change human behavior, not only to reduce disease, but actually save the planet.
Yes, I mean, agree, sir. That's a very well, if we could possibly do something in regards to that as well, I think that would definitely make sense. So as you mentioned, I think if we start on early, we could have the environmental factors helping us to possibly delay the symptoms, right?
Yes, that's right. I think that is the case. And that ultimately that's what we have in mind. And a broader point, following up my previous point, is that, many of the most common disorders affecting mankind and growing in many countries, like diabetes, heart disease, cancer, when you look at the lifestyle factors in these diseases, a lot of the lifestyle factors relate to diet, overeating, high fat, high sugar, but also lack of physical activity, sedentary behavior. And these all relate to behavior, whether it's, we think, and there's people at the Florey working on this, that a a lot of overweight and obesity is really addiction. It's food addiction. It's addiction to fat and sugar. So the core to overweight and obesity, we think, is a form of addiction, a form of food addiction. And the rest of it is failures of behavioral control in terms of, failure to eat the right food or the right amounts of food, but also failure to exercise adequately or do physical activity. And so these all come back to the brain and behavior. And so all of the most common lifestyle factors that affect all these common human disorders are driven by brain and behavior. And so, in the broadest sense, all of the major disorders that afflict mankind have their origins in brain and behavior.
Really interesting observation, sir. And the next question from Amartya is that if we can say that the epigenetic marks altered due to stress for mating is more pronouncedly inherited than other time period. In other words, he's just asking if altered epigenetic landscape time dependent, if it's just time dependent and reversible.
Yeah, look, these are very interesting questions. The very complex spatiotemporal control of epigenetic modifications, enormous complexity. Obviously, if you have 1 trillion cells in your body, they can all have different epigenomes effectively. That's the level of complexity we're dealing with. And then their epigenomes can be changed over time. There's another level of complexity. And so the intergenerational and transgenerational epigenetics is overlaid on the individual's environment or experience, changing their epigenetics during their lifetime, if you like, if you see that. So you do receive this kind of intergenerational epigenetic inheritance from your parents, but your own experience also changes your epigenetics from conception through in utero period, early life, and so on, adolescence, and so on. So there's a lot of complexity, and there's a lot of spatial and temporal components we need to be thinking about too.
All right, sir. And I think we've got a very interesting question from Stuti here that says that if from a public and planetary health perspective, how feasible could it be to combine epigenetic methods and principles to treat and prevent, as you mentioned, neurodegenerative disorders from a clinical perspective?
Yeah, absolutely. I think it's no doubt it's possible. I can tell you one area where epigenetics has advanced is cancer. And so there are various drugs, which is epigenetic drugs, which are being trialed in cancer. That's one area. And there are some drugs, like HDAC inhibitors that have been tried for, neurological and psychiatric disorders, not necessarily totally effective yet, but there are, increasingly drugs are being developed that have epigenetic effects, and they are being trialed in brain disorders as well. But a lot of them weren't mechanism-based drugs. I think we need a lot of new drugs and other therapies as well, obviously non-drug therapies like environmental interventions. There's potentially a whole range of different interventions that might be suitable for a particular disorder, coming back to the concept of precision medicine and personalized medicine based on scientific data and mechanism.
All right, sir. Yeah, I think that totally makes sense. And I think we've answered most of the questions here. And all in all, it was a really interesting conversation, and I really loved to talk, especially how the conversation steered towards certain important things and possibly towards the prevention of these diseases in the future generations, which I think again is truly important. And we had a great time. It was a pleasure having you here, totally. And okay, I'm really sorry. I think we have one last question that Aditi wants to ask: how far are we from reaching a point where we treat neurodegenerative disorders completely?
Well, yeah, how far? It's a difficult one always, people say 10 years or 20 years. There's a lot of different disorders, and it's, these clinical trials take a lot of money and a lot of time, and it's hard to stop them. I think ultimately, yes, I think eventually, the problem being, and this is where we all need to engage with our governments and politicians and others we need more funding for health and medical research. I think, if if the world spent as much on health and medical research as they spend on military and weapons and so on, then we'd have cured all of these diseases. You know, we're talking about the trillions that are spent on weapons and war and defense and so on. If that was all spent on health and medical research, we'd have cured every disease known to man. I know that. You know, so it's really a case of resources. If governments can spend more on health and medical research, then we will treat and cure all of these disorders faster. It's just a matter of time.
Agreed, sir. And I just hope that all of the countries reach a point where we do not have to wait for the funding to come in, so that we can all work towards improving these things. All right. So I think we have finally come to the end of the discussion. And as I said, it is totally a pleasure having you here. We want to thank you from the team, the entire team of NeuroNov, for having you here and, you know, giving us the time to speak and being open to these questions. However, do you feel comfortable putting in your email ID or perhaps a Twitter handle that you would like to take additional questions afterwards?
Sorry, what? I missed that question.
Sir, I was saying that would you be comfortable with possibly sending your email address or Twitter handle that other people could ask the leftover questions at?
Yeah, maybe the email address, I think probably Anashri and Stuti have my email address. So if someone has a specific question I haven't answered, then if you want to pass on my email address specifically to them, then that's fine.
All right, sir. Thank you so much. We can definitely do that. So thank